Perfect. Good evening and welcome to tonight's MBL Friday evening lecture. My name is Indu Sharma and I'm an associate professor in the Department of Biological Sciences at Hampton University. It's a historically Black college and university in Virginia, and I'm also a 2021 EE Just Fellow in the MBL Whitman Center. Thank you to those who have come out to Camellia Clap Auditorium and those who are joining us virtually. Throughout this summer, we are recognizing past members of the MBL community who have made extraordinary contributions to this storied institution. It is my privilege to share with you highlights of the life and work of Joel Plummer Cobb, an outstanding scientist and advocate for women and minorities in science, as well as an investigator, library reader, and corporation member here at the MBL. Joel Plummer Cobb was born in Chicago in 1924. Her father, Frank, was the first Black person to receive an MD from Cornell University. And her mother, Carrie Bell, also a college graduate, was a physical education and dance teacher. From a young age, they encouraged Joel towards intellectual exploration, including of her father's scientific library. An excellent student, Cobb originally planned to pursue physical education, but fell in love with biology as a sophomore in high school. In 1942, she gained admission to the University of Michigan, a major research university that had more than 200 Black students enrolled. Unusual diversity at that time. However, once there, Cobb encountered housing segregated by race and rampant social exclusions. After 3 semesters, she transferred to Talladega College, a historically Black college in Alabama. There, she found important mentors, including bacteriology professor James R. Hayden. Cobb pursued graduate studies at New York University and earned her PhD in cell physiology in 1950. Her doctoral research formed the basis for her lifelong interest in melanin and skin cancer. For her dissertation, Cobb characterized tyrosinase, an enzyme required for melanin synthesis, and tested its use for producing melanin in the lab. Later in her career, Cobb showed that deeper melanin pigmentation protects skin cells from X-ray treatments, providing the first evidence for melanin's ability to protect cells from damaging UV radiations. While a graduate student, Cobb spent the summers of 1949 as an independent investigator here at the MBL, studying the inhibition of cell division in sea urchin eggs. This was an early foray into a research interest Cobb would develop extensively in later years. How a hormone, ultraviolet light, and chemotherapeutic drugs can cause changes in cell divisions. Upon graduation, Cobb received and accepted 3 prestigious postdoctoral fellowships, one at Columbia University, one with the National Cancer Institute, and one at the National Cancer Institute at Harlem Hospital. At Harlem Hospital, she worked under Louis Tompkins Wright, the first Black graduate of Harvard Medical School, and established an important research collaboration with his daughter, oncologist Jane C. Wright. In 1952, Cobb founded her own laboratory at the University of Illinois Medical School, where she established the school's first tissue culture facility. 2 years later, she moved her lab back to Harlem Hospital, where she resumed her innovative collaboration with Jane Wright. Wright was studying the effects of chemotherapy in cancer patients, and in parallel, Cobb cultured cells from the tumors of these patients and studied their response to chemotherapy treatment. An early form of translational medicine, Cobb's results could be used to predict beneficial treatment for specific patients and cancer subtypes. Cobb and Wright made a critical contribution in the early 1960s by demonstrating the effectiveness of methotrexate in treating skin and lung cancer, as well as childhood leukemia. Methotrexate is now used for a broad range of cancer, including breast cancer and a lot of autoimmune disorders. Cobb went on to continue her cancer research as a biology professor at Sarah Lawrence College in New York. Then in 1969, she accepted an offer to become Dean of Arts and Science, as well as professor of zoology at the Connecticut College. This meant long hours, but it is also afforded Cobb a chance to nurture her growing desire to increase the marginal number of women and minorities in science. Cobb went right to work, establishing a Black scholarship at Connecticut College and a post-baccalaureate program for minorities to prepare them to pursue a degree in medicine and dentistry. Cobb became the first Black woman appointed to the National Science Board, helped form the first iteration of the National Science Foundation's Committee on Women and Minorities in Science, and chaired the first conference for minority women scientists. In 1976, Cobb became full-time dean of the Women's Division at Rutgers University and retired from laboratory research. Over the next decade, she researched and wrote numerous reports on the underrepresentation of women and minorities in science, and worked to institute academic programs to address these inequalities. In 1981, Cobb became the first Black woman to lead a college west of the Mississippi, where she was appointed president of California State University, Fullerton. Cobb's first summer at MBL led to a deep connection with Woods Hole, where she eventually brought a second home. She became an MBL Corporation member in 1972 and was a named and emeritus member of the MBL Society in 2007. She was a library reader for many years and also served on the MBL campaign steering committee. She died in 2017, and in 2020, Agassiz Road in Woods Hole was renamed Jewel Cobb Road in her honor. We are pleased to honor Jewel Plummer Cobb and her contributions to the Marine Biological Laboratory. She once said she had liked to be remembered as a Black woman scientist who cared very much about what happens to young folks, particularly women, going into science. It is our privilege to do so. And now I'd like to turn it over to Linda Hyman, Borough Welcome Director of Education at the MBL, who will introduce tonight's speaker.
Thank you, Indu, for that wonderful tribute. Now it is my great pleasure to introduce Dr. Shelly Berger as this evening's distinguished lecturer. In doing so, it's worth noting that it is more than fitting that she is following on the heels of the tribute of Dr. Cobb. They have some things in common, the least of which is their common university degrees at the University of Michigan. But perhaps more importantly, a passion for cancer research, a strong dedication to teaching and mentorship, and needless to say, a very distinguished career. So a bit about Shelley. She started her academic journey at the University of Michigan, then went to Harvard and MIT for her postdoctoral work, working with Matt Messelson and Lenny Garante. She then moved to the University of Pennsylvania for her faculty position, first at the Wistar Institute, and then in 2009 to the Departments of Cell and Developmental Biology, Genetics, and Biology, where she now holds the Daniel Oakes University Professorship. I have known Shelley for many years as a peer and as a friend. And it is from that perspective that I'd like to share why I think she is the ideal person to deliver this lecture tonight. As you know, Friday evening lectures are generally focused on exciting science, and I think you will agree that she will bring that to the podium this evening. Her pioneering and indeed her continuing work on epigenetics is transformative. It changed the way we think about the central dogma, DNA to RNA to proteins, and noted that phenotypic traits can be inherited without changes in DNA sequences. I'll let her tell you more about that. Her work in this area has been documented in more than 200 publications in all the prestigious one-name journals and beyond. Shelley is a fellow of the American Association for Advancement of Science. She is a member of the American Academy of Arts and Sciences and a member of the National Academy of Medicine and the National Academy of Sciences. There are no shortages of things I could tell about you in terms of her honor. But the real reason that I should say the other reason that I am delighted to welcome Shelley here is because I really believe that her work embodies the MBL spirit of fearless science. And what I mean by that is to work on the system that works for the questions you are asking. In that light, she has studied viruses, she has studied yeast, she has studied flies, she has studied mammalian cell cultures, and more recently, ants. And what I hear from her colleagues in that study, she's known as the Queen. Shelley also has a passion for science and discovery, which is what we all hold dearly. In fact, I'm going to share a quick personal story that sort of embodies how she can take that to an extreme. And that is, I recall visiting her in the hospital when she was about to give birth to her beautiful daughter, Ariel, while looking at autoradiographs of her latest experiments in between labor pains. I am not kidding. One might say that this takes the passion for science and that fire in the belly to new levels. With that, please join me in welcoming Dr. Berger this evening. She will share her work on epigenetics, not genetics, specifies complex behavior in ant societies. Shelley.
Okay, I didn't expect that little anecdote, but Linda, very, very cute. I don't know where you could have dreamed that one up. It's very true. It actually is not apocryphal. It's true. Something like that anyway. Oh, okay, I have to do something here. And let's make sure I do it right because I'm going to get yelled at by the MBL. See. I think I did it wrong. Start again. Got it. I got it. I think I passed the test. I might have. We'll see. No? Oh, this is hysterical. Sorry, you're gonna have to come down, Mike. He told me that the person who gave the talk last Friday couldn't do this. He said very disdainfully. So it looks like I'm in this, looks like next week's speaker is going to be told about me and my inability to do this. I did. Oh, I see. I just didn't. I was close.
There we go.
Okay, great. Okay, so Linda, thank you so much for that really overly kind introduction. Linda and I, I'm really in trouble. All right, right. It's not going very well. All right, so Linda and I do go back a long time to our years in Boston together, during postdoc, et cetera. Okay, so I'm going to go on and try to give this talk and not embarrass myself any farther. So today I'm going to talk, as Linda said, about ant societies, it's true, a real passion of mine. And my lab does study genetics and aging and mammalian learning and memory and epigenetic control of those areas. So the topic of ants is really quite a departure from those studies. All right, so let me get started then. Okay, that's not working. Okay, now it is. Okay, so let me start with an introduction, a pretty broad introduction, discussing the idea of societies. So humans, as we are all too aware, are among the most successful organisms on Earth. And this is widely thought by humans to result from cooperation amongst distinct individuals in societies. Societies being the aggregate of people living together in an ordered community. And this goes back, of course, to to prehistory and even to Neanderthals, I understand, of the cooperation between individuals to create societies, then through the amazing societies, the sophisticated society in Egypt, and then, of course, to today's society, and even the cooperation that is required to sustain these enormous cities that we have all over the world. Well, although we're all interested in what are the mechanisms underlying societies, we can't really study that in humans. And so we have to turn to other organisms. And ants turn out to be an amazing example of cooperativity, also culminating, like human society, in very sophisticated and complex societies. So here is a picture of an ant nest, and you can see the different morphology ants, and they're doing different tasks in the nest. So they have individual roles in the nest. And here you see the preeminent ant biologist of all time, E.O. Wilson, credited with at least discovering 500 species of ants. And here he is with the most wonderful leafcutter ants, models of the leafcutter ants in front of him. E.O. Wilson of Harvard, of course, again, really developed ant biology. He coined the term sociobiology, and I think it's widely accepted now, controversial in the day, widely accepted now that all biological systems, societies and biological systems share many attributes. And now he's a real advocate, of course, for species conservation. He does a great deal of work on that. Well, in this interview, I'd like to just share a few words from an interview I got online from the Sierra Club, a recent one, 2019, where the interviewer said, you've pointed out that species that work well together, ants and humans, are the species that have taken over the planet. And he responded that some explanation of that. Within groups, selfish members beat altruistic members on an individual level within groups. However, altruistic groups beat groups of selfish members. And he goes on to say, that's the driving force that has been important in the formation of us, of human societies. And it's the best trait of the human species. Okay, so that's very interesting. And I think we all have a sense of what altruism means, a very good sense of what altruism means for humans. But what does altruism mean for ants? You know, here we're going to talk about ants as a model for societies. So reproducing oneself is a tremendous motivator in all of biology. We all know that. Many of us are scientists. We know that that's an imperative that drives biology. However, individual ants forego reproduction and instead become dedicated workers in the colony. This system of the queen carrying out all the reproduction for the colony and workers doing all other tasks is called a caste system. That's come to be known as that. Okay, so now let's take a look at some ants here in this slide. So ants have evolved. Ants span hundreds of millions of years of evolution, and they've evolved increasingly complex societies. Here are 3 ants that we study in my laboratory. Let me introduce them to you. On the left is a more ancestral, primitive system of ants. I'll show you that in a second in a little bit more detail. I'm sorry, these are 3D renderings prepared by a postdoc in my lab, Belinda Kecksaw, and a graduate student, Michael Gilbert. So these beautiful renderings of the queen, and I'm going to go through this in a little more detail in a second. So here's the ancestral system. Here's an intermediate system of a few more individuals. And then here's an advanced caste system, the leafcutter ant. So let me then go through this in a little more detail with some videos to show you what these ants look like. So here on the left then again is Harbignathus saltator. It's a primitive ant system. There's a queen, there's queen and worker. They are morphologically very similar, and a male. And the queen, and it's really only 2 to 3 caste members, caste types. And if the queen is removed from the colony, you can see that there's dueling between worker ants. And then one or more of them can emerge now as a queen, as a reproductive. Okay, so that's a primitive ant, only 2 to 3 castes, different castes. Here's an intermediate caste system, Camponotus floridanus. There's a smaller one called minor, a larger one called major, male and queen. You can see these colonies are much bigger. There are, as I said, 4 castes. And then on the right is the Atta cephalotes. It's an advanced caste system, leafcutter. There are 10 different castes of leafcutter ants. You can see our colony in the lab, and the supermajors, these very large supermajors walking around, and other ants carrying leaves and carrying out the tasks of a colony. So these are the different examples of different ant societies over evolutionary time. Okay, so we think of human destiny as occurring via genetics, environment, choice. But how do these lowly ants, how do they fulfill their caste roles? So you've just seen the dramatically different shape and behavior of these ants. And what's amazing is that they arise from the same genome. So hence, it is epigenetic, not genetic differences between individuals that determines ant destiny. So what is epigenetics? Let me define that for you. Epi means on top of genetics, on top of genes. So let's go through what this means, I think in a simple, intuitive way. So we all begin life as one cell, a zygote, union of a sperm and an egg, and then go through development to a fetus. And then there are at least 200 or more different cell types that emerge from this initial single cell. So epigenetics, identical genome by definition, all from one cell, one genome, and 200 different phenotypes or cell types emerge. All right, so the word epigenetics was coined by Conrad Waddington in the 1950s, 1960s. He was a visionary scientist. He coined this idea of an epigenetic landscape, drew this amazing diagram, and the idea that development in a multicellular organism is defined by this landscape of the trajectory of this initial single cell through development and it becoming the multicellular organism. So again, one genome, many different phenotypes. And so we can place now on this Waddington landscape the idea of one genome, a stem cell, becoming many different phenotypes, all the different cell types of our bodies. So epigenetic regulation then is central to establish and maintain cellular identity. All right, so now with the ants, we can place them as whole organisms on the same Waddington landscape. Here, from single genotype embryos can differentiate these different castes, the male queen and the different types of workers. And indeed, these eusocial insects live in colonies that are really analogous, if you will, to differentiated organisms. And further, E.O. Wilson himself, with his great collaborator Bert Holdeber, called this colony the superorganism. So this is our organism, and the colony is the organism of the ants, with all the differentiated individuals combining to the society, to the whole. Okay, so epigenetics in society. These individuals are genetically identical. And so epigenetic mechanisms must toggle between, or I guess the question is, is it then epigenetic mechanisms that toggle between castes for these behaviors to emerge, for these individuals with different behaviors to emerge? All right, so let me very quickly and painlessly go through a simple primer in epigenetics. As we're aware, inside the eukaryotic nucleus is an enormous amount of DNA. 3 meters of DNA in every cell has to be packed into this nucleus, and it does so. The double helix is packaged around packaging proteins called histone proteins forming nucleosomes, these individual units of packaging, which then form the higher order structures that then get packed into the eukaryotic nucleus. All right, so there are many different epigenetic chromatin mechanisms, but we're only going to talk about really one of them. I'll start with the one that many of you have heard of, DNA methylation, direct little chemical methyl groups that are covalently placed onto DNA. So that's a well-known epigenetic mechanism. The histone proteins also become modified with little chemical groups. And the only one we're going to talk about today is the acetyl group, a small chemical group that's covalently placed onto histone proteins to regulate them. All right, so what does acetylation do? It opens genes to turn them on. So here on the left, these little cylinders represent the nucleosomes that you saw on the last slide. And these little green balls are the acetyl group. So acetylation is placed by enzymes called histone acetyltransferases, or HATs, and that opens up genes to turn genes on. And this can be reversed by deacetylation, enzymes called histone deacetyltransferases, which then close up the fiber, the DNA fiber, and to turn genes off. All right, so that's the introduction, painless, user-friendly on epigenetics. All right, so now we're going to start talking about a little data in the lab using one of our laboratory models that you saw on an earlier slide, the carpenter ant, the Florida carpenter ant, Camponotus floridonus. So this species then has a queen and two types of workers. The smaller called minor worker, that is the forager. This worker goes out and forages for food and brings it back to the colony, to the nest. And on the left, the major worker, much larger, you see the big head, big strong mandibles. This is the soldier in the colony. All right, so let me test you on your knowledge of ant biology. True or false? Or these are all, here we have the female and here we have the males. Is that true or false? False, great. They are all females. So the workers in the colony and the queen are all females. The males, sorry guys in the room, the males live about 2 months and they basically supply sperm to the queen. And then they fulfill their destiny, and that's about it. Actually, this is an amazing aging model as well, because the queen in this species can live 30 years. These workers live about a year, and the male, as I said, lives about 2 months. So we also study ants for aging. Questions of what are the epigenetic regulatory mechanisms of aging. Okay, so we're going to study caste behavior, though, today. We're going to discuss caste behavior, not aging. So again, the minor ant on top is the forager. The major ant below is defense. And what we discovered now almost 10 years ago is that a single type of that histone acetylation, that little green ball, there are different types of those balls, a single type strongly correlates with caste identity. So we published this some years ago, and that was pretty interesting, that this one type of acetylation was important to at least correlating with caste identity, because even to this time, it was known in neuroepigenetics in mammals that histone acetylation is crucial in learning and memory. Histone acetylation enzymes are seen to be mutated in certain intellectual disability syndromes. So that was pretty interesting. So that led to the first, I think, really interesting set of experiments that we did with the ants in behavior. We asked, can we alter caste-specific behavior by changing histone acetylation? Specifically, can we convert the major, the soldier, to foraging? And this is work done by a postdoc in the lab, Dan Simola, with a graduate student, Riley Graham, and published some years ago now. Okay, so now you saw this earlier in the video. The carpenter ant lives in large nests. This is a medium-sized nest. They can go to many, many, many thousands of ants. And you can see we can mark them with paint, so we know what age they are. But you can't really study behavior in this, in this lab nest. It's just, there's too much going on. You can't track behavior, et cetera. So we needed an assay. And so we developed an assay to assay foraging, and that's shown here on the right. So the red mylar is where the ants are living. They don't see red light. They live, that's their nest. And they come out, go through this Tygon tubing over to this foraging arena with some sugar water. That's they're foraging for sugar water. The scout, a really smart forager, can come back to, samples the water, the sugar water comes back to the nest, shares it with the others, and then a bunch of foragers come out and they're drinking buddies at the sugar water. Okay, so that's the assay. Now we can track the ants and quantify their foraging, the rate of foraging. All right, so now we're going to have to you do something to alter the acetylation level. And what we did a number of things, but one thing we did was use drugs to alter acetylation levels. So histone acetyltransferase is put on acetylation. HAT inhibitors then lead to less acetylation, makes sense. Histone deacetylases remove acetylation. Histone deacetylase inhibitors lead to more acetylation. So we can toggle back and forth between higher and lower levels of acetylation. So we then use 1 day old, newly hatched, newly closed majors and inject into their brain a single dose of inhibitor, HAT inhibitor or HDAC inhibitor, and we can follow it with a fluorophore in the solution that we inject. And now we can then assay foraging rate in the little assay that I showed you before. And this is again done in majors. So DMSO is the control. And you can see that in a dose-dependent way, with HDAC inhibitor, increased acetylation, we can increase the foraging rate. If we now add in, I hope you can see that, everyone. If we now add in HAT inhibitor, we can now, at this level of HDAC inhibitor, we can, in a dose-dependent way, lower the foraging rate. So that's quite interesting. So that leads to my first summary. The HDAC inhibitor, which increases acetylation, induces foraging in majors. This foraging reprogramming, I didn't show this to you, but it's sustained for weeks following the injection, in spite of the fact that the drug lasts only hours. The behavior then is altered permanently, and so that is the hallmark of something called epigenetic memory, which is sort of the gold standard in epigenetics to show that an alteration is sustained for a long time. Now you know you've done something that's carried on through cell division, et cetera. All right, so that led to a lot of interesting questions. Very interesting questions, both with regard to the reprogramming of the major soldier to foraging and natural programming of the minor, who's the natural forager. So here are some of the questions we became interested in. How long is this window of vulnerability to reprogramming? What is the mechanism? Is it truly epigenetic? And what about natural programming of cast identity to minor? Are these mechanisms related, the reprogramming and the natural programming? And this work was published last year, led by Carl Glastad, a postdoc in the lab, with Riley Graham, a graduate student in the lab. Okay, so this was our approach to address the question of the relationship between reprogramming and programming and potential epigenetic mechanisms. First, we determined, you'll see in a second, the optimal window of reprogramming. Second, we find genes that are similarly expressed during reprogramming of majors to foraging and natural programming of foraging minors. And third, we investigate the mechanisms, asking, is there an epigenetic switch? Okay, so here you're looking at data recapitulating what we saw earlier, injection of control or HDAC inhibitor into the brain of a day 0 major. And what you can see, just as we saw before, an increase in foraging, an uptick of foraging. Okay, so now we look later. We look at day 5 after eclosion, after hatching, or day 10. And I think you can see in a glance that there's a much bigger effect at day 5. This is a much more substantial effect, increase in foraging at day 5 injection. And very interestingly, at day 10, you can see the effect is gone. The window has closed. So we've defined the window of susceptibility to reprogramming in majors. And that reprogramming is highest at 5 days and then closes somewhere between 5 and 10 days. We don't know where yet. Well, this is pretty fascinating if we start thinking about ant versus human again, going back to the original questions related to human learning and brain plasticity, because we know that very early in life, for example, children are able to learn languages with great ease, but that window of plasticity closes as the children age. All right, so that's interesting. Second question then, what is the mechanistic pathway of reprogramming? All right, I have to give you another factoid to follow the next part of the talk. Foraging requires hormones in ants, just as many of our behaviors require hormones. Foraging requires a hormone called juvenile hormone, well known in the insect world. Juvenile hormone is degraded by enzymes called juvenile hormone esterase and juvenile hormone esterase hypoxide, I think. So two different enzymes degrade juvenile hormone. Okay, so now we carry out RNA-seq, and we do that at day 5, where we get this optimal HDAC injection reprogramming that I showed you in the slide a moment ago. Okay, and we depict the altered RNAs in this typical type of volcano plot, where on the right are the genes that are going up in expression, and on the left are the genes that are going down in expression. And I think you can see in a glance that there are many more genes going up than down. And this makes sense because the HDAC inhibitor increases acetylation, which is largely turning genes on. So that makes sense. Okay, so what do we find here that might help us to start to unravel the mechanism? Well, on the right in the genes that are going up, we spotted, there are a lot of things, but we spotted a complex that we thought was really interesting. That complex is composed of a transcription factor called TRAM-TRAC in flies that we've named the same in ants. And TRAM-TRAC is a DNA binding transcription factor, and it is a repressor. And it's a repressor, and it recruits to DNA to turn genes off a co-repressor complex, and we saw an upregulation of that complex. One component of that complex is called co-rest, and it's a very large scaffolding, what we call a co-repressor scaffolding protein, and it brings in enzymes that are actively repressed via repressing the epigenetic chromatin fiber, such as upregulation of an HDAC. So that you can see that was very highly upregulated. So we have an epigenetic enzyme repressor, an epigenetic co-repressor, and a DNA binding repressor, all going up. Okay, so that's interesting. What's going down? And we saw in going down this juvenile hormone esterase, this juvenile hormone degrading enzyme. All right, so before I get to what we think is going on, let me go on to a couple more things that we observed. We wanted to ask again, is this mechanism of reprogramming in the majors similar to the natural programming of the foraging minor? So here we again do look at what genes are upregulated now, either in minor on the left or major on the right. Upregulated genes in the minor, amazing, co-rest and TRAM-TRAC again, upregulated in the minor. That's foraging. And in the major upregulated are these two juvenile hormone degrading enzymes. Okay, and one more thing, I'm not going to show you the data, but we carried out genetic lowering of co-rest, and we found that it's crucial for major reprogramming. So that's pretty amazing because the knockdown of a single gene, even though you saw all those genes going up, a knockdown of one single gene led to it no longer reprogrammed to foraging. All right, so here's what we think is going on. I'm going to just summarize a lot of data. So we have a hypothesized circuit for foraging. Juvenile hormone promotes foraging in minors, and then juvenile hormone, these degrading enzymes degrade it, and it activates juvenile hormone in majors. Okay, and more specifically, the mechanism we think that's going on is that TRAM-TRAC is recruiting co-rest and HDACs to turn off these degrading enzymes. So this is what's happening in programmed minor and reprogrammed major. This leads to a higher level of juvenile hormone and foraging. In contrast, in the normal major, we think that these genes are on. We don't know the transcription factor in the hat yet, but these genes are on, and juvenile hormone levels are low, and there's no foraging. Okay, So let me summarize then. Caste specification initiates early in life in minors via tram-track and CoREST repression of juvenile hormone degrading enzymes, resulting in a high level of juvenile hormone, and reprogrammed majors also show a high level of juvenile hormone. So they're tapping into the same mechanism. All right, so what is the significance to human behavior by all of this? Well, again, we think this window of ability to be reprogrammed shows early life plasticity. And I'll point out that these ants by 10 days are kind of getting to be real adults. So even in humans, we know that into late adolescence, it's been shown by sociologists that that the brain continues to be plastic in humans. So we think this window of plasticity is a kind of a reflection of possibly a similar phenomenon. It also reinforces the utility of the ant model that we're able to see this sort of window of plasticity early in life. All right, so let me in this last little section show you some of our current studies. Here we're focusing on how does this mechanism distinguish major and minor brains at the cellular level? How does this happen? And this is work carried out by a graduate student in the lab, Lin Yang Zhu, along with a postdoc in the lab, Carl Glastad. And this work is in review right now. All right, so I'm going to jump to some newer technology that many of the scientists in the audience know well about, and this is single-cell RNA sequencing. So here we're carrying out single-cell RNA sequencing of the two castes, major and minor, looking at brain heterogeneity. Okay, so I'm just going to cut to one of the key observations we've made. We made many observations, but one of the key observations is that juvenile hormone esterase, this degrading enzyme, its levels map to one very specific cell type in the brain, and that's called surface glia. And here you can see in the UMAP, in the trajectory, in the trajectory of all the different cell types, neurons, all kinds of different glial cells in the brain, you can see this one specific surface glial cell type, SG2. And what we noticed here is that the levels of juvenile hormone in this SG2 cluster of cells, the surface glia, are higher in major than in minor. First of all, I buried the lead, one of the leads here. Juvenile hormone esterase maps very specifically to this cell type, and it's higher in major than it is in minor. Indeed, if we look at these violin plots of the levels of juvenile hormone in major compared to minor, you can see a much higher level in the major. So the levels are much higher in major than in minor in these surface glial cells. All right, so here is our hypothesis for what's going on. I'm going to cut right to a schematic. Our hypothesis is that juvenile hormone esterase is expressed in the surface glia of the cells to regulate juvenile hormone levels in the brain. So here's a little schematic. Here's the outside of the brain where the blood is, the insect blood called hemolymph is flowing around the entire surface of the brain. They have an open circulatory system. And here's juvenile hormone outside of the brain. Here's the blood-brain barrier, the equivalent of the blood-brain barrier in the insect, the surface glia. And here is where juvenile hormone esterase is localized. And it's then serving as a gatekeeper, degrading juvenile hormone before it can reach the interior of the brain. So the blood-brain barrier is functioning as a gatekeeper to dynamically regulate juvenile hormone levels inside the brain and regulating foraging behavior. All right, so we've done many, many different tasks. I can tell you more about it if you like in the question and answers, but right now I'm only going to I'm going to show you one additional experiment. And this is a spatial expression of juvenile hormone in the ant surface glia. So this is immunofluorescence staining and confocal microscopy that you're looking at of the whole brain of the major versus the whole brain of the minor. DAPI staining the DNA in the nucleus, synapsin antibodies staining the neurons, and JHE antibody staining for juvenile hormone esterase. And I think what you can see at a glance in the whole brain and in these higher magnification insets is that the expression of juvenile hormone esterase is very specifically in the surface glia around the brain and much higher in major compared to minor in this view. So the expression of JHE occurs in the surface glia much higher in the major ants. All right, so let me then summarize what we think is going on during natural caste-specific programming of behavior. So in the major ant, where juvenile hormone, where the esterase, the degrading enzyme, is highly expressed, there's lots of it in the blood-brain blood-brain barrier, and so it's breaking down most of the juvenile hormone before it can reach the interior of the brain. So there are very low juvenile hormone levels inside the brain and low foraging. What's happening in the minor? We have the repressive complex, tram-track and co-rest, lowering the levels of juvenile hormone esterase, much lower levels in the blood-brain barrier. So much less of it is broken down and much more of it then reaches the interior brain, higher juvenile hormone levels inside the brain, and high foraging. And I'll just then summarize everything I've shown you today, because what we think then is the HDAC inhibitor injection then lowers co-rest, increases co-rest and tram-track, decreases juvenile hormone esterase, increasing juvenile hormone, and that co-rest knockdown blocks this reprogramming. All right, so one last piece of data that we think is pretty interesting. We've done some computational searches of hormone-degrading enzymes in the blood-brain barrier in mammals. And we find examples, androgen and estrogen metabolic enzymes that are high in brain endothelial cells, that are the cells that are in the blood-brain barrier, much lower levels in the whole brain in all cases, and lower levels in other endothelial cells from other organs. So that leads us to think that this blood-brain barrier-based hormonal degradation may be a general mechanism to regulate brain hormone levels, and this is an entirely novel mechanism then that may be conserved. All right, so then to summarize, ant workers have identical genomes, but dramatically different phenotypes. Epigenetic regulation, we believe, controls social behaviors or aspects of social behavior that we've studied. And understanding epigenetic regulation in ants, we believe, will illuminate behavior in many systems, including in humans. And then finally, on a lighter note, Carl, this postdoc you keep hearing me talk about, he's quite amazing scientist, is also a pretty cool cartoonist, as you can see here. Here's the minor forager going out to collect crickets and other types of food items. Here's the major soldier, big and bulky and ready to do battle with her spears. And then here's the reprogrammed soldier, the big mighty soldier now helping to carry out foraging. All right, and with that then, my final slides. Here are all of these terrific postdocs and students that you've heard me talk about in the lab. And Dan Samola is a principal investigator at GlaxoSmithKline. Riley Graham is a principal investigator at GeneWiz. The others are still in the lab. Here's our funding, and I want to shout out to HHMI, who funded the beginnings of this project, the sequencing of our first two ant species. They funded us twice through a collaborative Investigator Award, and us means Danny Reinberg at NYU. Danny and I really started these epigenetic type projects together with Jürgen Liebig at Arizona State, Claude Desplan at NYU, and Roberto Bonacio now at Penn. We've also have funding on our ANT studies from National Institute of Aging at NIH. They see the ants as a great system, model system for studying aging. And we have fellowships from NIGMS and from Damon Runyon because Balint is trying to stand up the ant system as a system to study social effects on cancer. And here's my lab shown here in another picture, a more relaxed moment. And I know there are a lot of males in our ant project. I guess little boys love ants, little girls too, like me. But I do have lots and lots of women in my lab. I'm very proud about that, and many diversity trainees as well. With that, I'd love to take questions. And thank you very much for listening. Yes.
So just raise your hands, wait for the mic, and then we can take questions online as well.
Great talk. I'm curious about one thing. So I'm following you correctly. You add the age deck inhibitor.
Sorry, slow down a little.
Great talk. You show that when you add the age deck inhibitor, you see then these genes are go up in expression.
Mhm.
Have you looked at their epigenetic states? Like, I'm particularly curious, is CoREST already on? So it's a matter of levels changing, or is it actually off?
Yeah.
What does that look like?
Yeah, great question. So we've now gone back. We've done a lot of work on this now, and we've gone back into the pupil stage, and we see it getting turned on during the pupil stage. So we think the programming is happening quite early in development. I should say it could be on some of this could be on even earlier in larval stages, and we're doing that now. So we're going way back into earlier development to try to look at the underpinnings of this. We also are, we've carried out a lot of ATAC-seq to look at opening and closing of the genome, and we do see opening. We're not quite sure what's regulating CoREST. So thank you very much. And if any of my postdocs or students are online listening, did you hear that question? Any other questions? Yeah.
So do you know what happens when the reprogramming window is closing?
Great question. They're listening, I hope, because I wrote on an email just before I came on the ferry coming over from Martha's Vineyard to one of my postdocs. Remind me, do we have data on when the window is closing? I don't know yet. I think it's a great question because that's going to be a really interesting epigenetic, another repressive type mechanism probably to close up these windows.
We have one in the audience here, and then we'll go to one online.
Hi, so you mentioned that there were behavioral changes. Did you ever notice any morphological changes, or if those are epigenetically regulated in any way?
Right, great question. By the time they close or are hatch, they're already morphologically mature. So they look like majors their entire lifetime after that. So they remain the same. And we haven't, so it's a great question, and we could go earlier in development and try to make it manipulate. You can see pretty early where they start to diverge, even in the larval stage or early pupil stage. And we've thought about trying to reprogram then. The leaf cutters, which are our newest species in the lab, they have an open pupil state. They don't have a cocoon around them. And so it'd be much easier to do that kind of reprogramming in the leaf cutters. The downside of the leaf cutters is that we have 10 casts. So, but we're finding ways to simplify that system to ask questions like this.
I think we have one online.
Could epigenetic mechanisms be invoked in other model systems like bees and wasps, where food and mechanical vibrations induce different casts?
Yes, absolutely. And so it's a great question. You all are aware of royal jelly. You know, it's the substance that's fed to larval bees to lead to queen development. And the really interesting thing about royal jelly, another lab, Mark Bedford's lab at MD Anderson, they took a look at royal jelly. They did a mass spectrometry on it and saw that there's a HDAC inhibitor substance within royal jelly. And indeed, when they use royal jelly, they can see that it has HDAC inhibitor capabilities. So really interesting. What is HDAC inhibitors remind you of? I showed you that we could change cast trajectory with HDAC inhibitors. So probably activating a lot of genes during bee development. So that's a great question. We are in ants, it's not quite as clear what the nutritional inputs are for cast differences. It's thought to be, new queens are thought to emerge, at least for Camponotus floridonus down on the Florida Key archipelago, by changes in barometric pressure because of spring thunderstorms, and that leads to new queens being made, and they then fly out. They're winged, fly out, and establish new colonies. So we don't know all the inputs. It's a great question, though. All the inputs into mechanical stimulation is also interesting. That was mentioned. All very interesting question.
I'm curious about the drugs that you used to change histone acetylation. Where do they come from? Are there anything like them that occur in nature that have a similar effect?
Well, I just mentioned one, the HDAC inhibitor that's found in royal jelly. That's a natural substance in royal jelly. So that's a really amazing example of a natural one. So butyrate, which is produced by microorganisms in our gut, is an HDAC inhibitor. It's a natural inhibitor, and sodium butyrate is used as an HDAC inhibitor in the laboratory. So that's another natural substance that is thought to lead to acetylation, to changes in acetylation metabolically in organisms. HAT inhibitors, I don't know of any naturally occurring ones. Those are developed in pharmaceutical and biotech. There are lots of these types of inhibitors around now.
So we have one more from our virtual
Okay.
audience.
Everything you described is behavioral, but the casts are morphologically distinct as well. Is that regulated epigenetically?
Yes, that question was asked previously. We can't manipulate that because by the time they close, they're already morphologically mature. But I'm sure that the morphological changes are also epigenetic back during earlier development, and we'd like to investigate those. So good, very good thought and question. Bob, yeah.
Shelley, maybe I take this one. You,
There you are. Good to see you.
can I ask if the modifications that you impose with drugs require a round of cell division, or can they be any interface cell can be altered in terms of
Right.
histone modification?
So this question has to do with DNA methylation. So some of the DNA methylation drugs require 5-azacitidine, famously, has to be intercalated into DNA during cell replication. And so let's say you're trying to do experiments in the brain with post-mitotic neurons, you wouldn't be able to get 5-azacitidine in. I will say that there are newer classes of DNA methyltransferase inhibitors that are inhibiting the enzymes, so you might be able to do that. The acetyltransferase, that's all done, that can all affect post-mitotic. So they do not have to be going through replication for HATs and HDACs to regulate. It's very fast. It's fast on and fast off. And that's why in the earlier part of the work, we were, it's amazing that a pulse of drug can lead to these long-lasting behavioral changes. So that means that there are other things that are happening downstream. Maybe DNA methylation, which is more of an epigenetic memory, maybe other histone modifications like methylation that could be more long-lasting. So that's a great point.
And one last one in the audience.
Do you think if you could balance out the concentration of juvenile hormone esterase and juvenile hormones together, would they be performing both sort of tasks in the caste system?
Huh. Okay, that's an interesting thought I hadn't thought about. So if we had high levels of juvenile hormone and I don't know. It's a good question. And I think your question really gets to the question of what's regulating major soldier behavior. And we don't know. We haven't really started to investigate that. Can we transition a forager into a soldier? So I think that's kind of getting at your question. We haven't tried to do that yet. It might be that increases of juvenile hormone esterase, the degrading enzymes might be able to do that. So that's a great thought. So yeah, thank you for that interesting question.
Thank Dr. Berger.
Okay, thank you all. Great questions. Really appreciate that.